Okay, this one is probably going to be quite dense, so we might move a bit fast. If any of the stuff I say isn't massively clear, I'd just recommend reading the slides, because they contain all the information that you really need to know. This one mainly going to be focusing on the different kinds of T cells, so the cytotoxic and the helper T cells, as well as the other kind of lymphoid cell that we mentioned at the beginning had a role in the innate immune system rather than the adaptive immune system, the natural killer cells, which are a distinct lymphoid lineage separate to the T cells and the B cells, but most of this is just going to be focused on the T cells. So, cell-mediated immunity is the main effector mechanism which T cells use to destroy pathogenic microbes. The cytotoxic T cells, with their T cell receptor and CD8 co-receptor, eliminate intracellular pathogens detected on MHC class I molecules of antigen-presenting cells. Meanwhile, CD4, so helper T cells with the T cell receptor and CD4 co-receptor, have a different function depending on what kind of helper T cell it is. Th1 cells detect intracellular pathogens. Meanwhile, Th2 and Th17 helper T cells detect extracellular pathogens. So Th1 they're both T cells with T cell receptor CD4 co-receptor, but Th1 is recognizing intracellular pathogens for the cellular immune response. Meanwhile, Th2 and Th17 detect extracellular pathogens for the humoral response. We're going to go deeper into the different kinds of helper T cells in a minute and their differentiation. Then we have regulatory T cells, which can either have the CD4, CD8 co-receptor, and they just regulate these responses to prevent too much damage to our own tissues. So both CD4 and CD8 T cells are activated upon binding of an antigen to their T cell receptor. Only T cells with a variable region on the T cell receptor that's complementary to the shape of the antigen presented on the antigen presenting cell will be activated. So once these T cells with the complementary T cell receptor are activated, they proliferate and produce millions of T cells from their lineage with that complementary T cell receptor that can recognize a specific antigen. So this proliferation is triggered by the cytokine interleukin-2 secreted by activated CD4 and CD8 T cells. I just wanted to highlight that quickly. So basically, when we have these naive T cells shown on the image on the right, they're a bit cut off, but you can see that the first cell, the white one, is the naive T cell. It encounters an antigen, mainly focused on the bottom one on the image on the right. It encounters an antigen. They then undergo clonal expansion. So just imagine here, it's dividing into multiple of these intermediary cells. And then once clonal expansion occurs, these naive T cells, they differentiate. When the infection is over, most of these T cells die via apoptosis, but some of them remain as memory T cells, and these memory T cells can either be the helper T cells or the cytotoxic T cells. They've already encountered their antigen, so they're basically stored as a precaution to re-exposure to the same pathogen. So if the same pathogen infects again, they can pick up the antigens and stimulate their own kind of immune response. Antigen presentations T cells are antigen presenting cells in the peripheral tissues, like we talked about in the last one, or dendritic cells or macrophages. They encounter the antigens in these peripheral tissues, then migrate through the lymphatic vessels, and they get to the lymph nodes. In these lymph nodes, they present the antigen to naive T cells. Upon presentation, these naive T cells are activated and proliferate via clonal expansion, like we talked about here. This can occur in the lymph node when antigen-presenting cells present antigens to these naive T cells, or it can happen if the naive T cells are just wandering about throughout the body in the lymph vessels or wherever, or in the tissues. These replicated T cells are all capable of recognizing the antigen on their T cell receptor because the clonal expansion will only occur if the naive T cells have a T cell receptor that recognizes the specific antigen. So if it has a variable region with a complementary shape, so they either kill infected cells if the pathogen is intracellular, if the T cells are CD8 cytotoxic T cells or Th1 helper T cells. But if the pathogen is extracellular, so like think larger bacteria or parasites or helminths or whatever you want to talk about, they activate B cells to produce antibodies that opsonize these pathogens. So as well as antigens, antigen-presenting cells also present things called co-stimulatory molecules needed for T-cell activation. But an interesting thing is that some antigen-presenting cells, like cancer cells, can avoid detection by T cells that would kill them. So you can imagine a cancer cell, it's presenting if a cytotoxic T cell recognizes that a cell is a cancer cell, it's going to want to kill it, but the cancer cells don't want to be killed, so these cancer cells get around that by producing their own co-stimulatory molecules, like PD-1 or CTLA-4, which lead to T cell inactivation, so the cancer cells are able to avoid being wiped out by this antigen presentation to T cell process. So an activated naive T cell with T cell receptor and the CD4 co-receptor is always going to produce a helper T cell. These are three of the main helper T cells we're going to talk about. If the naive T cell is activated with interleukin 12, it'll become a Th1 cell. If it's activated with interleukin 4, it'll become a Th2 cell, and if it's activated with interleukin 1 beta, it'll become a Th17 cell. And each of these different kinds of helper T cell produce their own kind of cytokines. So we can see here Th1 is going to produce interferon gamma, and we're going to go deeper into this probably in the next video now because we're running out of time. But like you can see, I just make a note of all the different cytokines that each of these different helper T cell types produce, and we're going to talk about what they do in the next one. Th1 helper T cells are mainly focused in getting rid of intracellular pathogens like we talked about before, so they're in charge of the cellular immune response. So they're going to be targeting viruses or mycobacteria which infect cells internally and proliferate within cells. Meanwhile, Th2 and Th17 both target extracellular pathogens that proliferate outside of cells, so like larger bacteria or parasites like helminths. But because they produce different cytokines, they target different kinds of extracellular pathogens. Like Th2 mainly targets parasites, meanwhile Th17 is mainly targeting bacteria and fungi. So yeah, we're going to go deeper into these different kinds of helper T cells in the next one.